My name is Vipat Roxakultai. I'm an application scientist for molecular devices. I support our high content imaging products, and I'm going to be talking about 3D imaging of cancerous spheroids, some developments that we've done in collaboration with another company to enable imaging of your spheroids in a higher throughput manner. All right. So I just want to discuss really quickly about the two different technologies right now for putting spheroids in your microplates for higher throughput imaging. Of course, you can put them just in Matrigel in a regular 96 well plate, but a couple of these technologies, one is the hanging drop system, and so what that is you have a suspension and your cells grow in the center of that, right? You can see on the top. And then on the bottom, you could photo treat and these microplates so that they promote spheroid formation in an arrayed format to make them easier for imaging and analysis. I'm going to talk about the second on the bottom with work that we've done with a company called Sellable from Transparent out of Japan. So what you're seeing here is a zoomed in version of the plate. So within each well, there are about 100 patterns here that have been photo treated. And once cells are seated inside the wells, they migrate to these arrays and start to form spheroids after about 24 to 48 hours. We can seed you can put the plates inside the imager, and what we're showing is the ImageXpress Micro XL, and it can automatically acquire these plates because they're in a 3D 4-well format in multiple colors, up to 5. And a 3D 4-well plate, you can do it within in a little bit less than 15 to 20 minutes or so. Right? And there's a lot of literature out right now just showing the differences between 3D cell culture systems and traditional monolayers, traditionally plated cells. On the top, what you have is a traditional 2D cell culture system compared to the bottom of the 3D, so within the sellable plates. And you can see that when we treat these cells with a compound, then we'll be able to see the differences in between the two culture systems. The 3D models are more resistant to chemotherapeutics. Okay. And then also another study showing the results between the difference of size between small spheroids on the left and large spheroids on the right. You may not be able to make out the words here. So this was a necrosis and apoptosis assay. The coloring here is blue is for the nuclear stain, the DAPI, the cow, so that's the apoptotic stain, and then ethidium heterodimer here in red. And just showing between the vehicle control and when you treat it, with a control compound, the size of the spheroid, as you can see, once we have a large spheroid, these make them more resistant to drug treatment than when you have a smaller spheroid size. So what helps as well with the plate type is that you get more uniform examples of your cells. Okay. Whoops. Okay. And then just another example showing a couple different dose treatments of these DLD1 cells in response to two different compound treatments in both nuclear intensity and also EDU uptake, so that's a marker for proliferation. And then these two movies are just showing the time lapse. So one thing, instead of just traditional endpoint assays that we're seeing, is more interest in performing live cell assays, monitoring your cells over time. That way you're getting a single well, but lots of data, multiple data points within the single well. So I'm not sure if that played as well. So this is the color merge movie of the three different stains here. So green is the viable cells, and then red, these are our necrotic cells. And then when you compare them in time lapse, so you'll get a lot more information, just for the same cells. We're monitoring these same cells over time. And this also plays in well with some capabilities of the hardware, which I'll go into towards the end of the presentation. Okay. And then just showing some, starting to talk a little bit about the analysis. One analysis that, simple analysis that is requested is also just monitoring spheroid size and area. And this is just showing an example with a white light, a non-labeled sample. We wanted to get the size and area of this structure. And using the software that's coupled with the ImageXpress Micro, we're able to get multiple parameters out. So it's total area, perimeter, and length and width, various parameters for this structure here. Right. So taking it back a little bit, I've talked about within a plate, multiple plates, and for high throughput, if you wanted to do some more development work and use the instrument more in a research mode, I'm going to outline a kind of a workflow for that if you wanted to maybe just study less samples but get a little bit more information. In this case, we're looking at a 96 well plate, imaging at a 2x in bright field. What we wanted to do was just identify. They're just in a regular 96 well, or 48 well plate, I'm sorry. And we just wanted to identify our spheroids, our objects of interest, and then move to a higher magnification so we could use the instrument more of like a standard upright microscope in this case. So what we're showing is that we selected our regions, our objects of interest, and we wanted to investigate them further. And the previous data I showed was at a lower magnification, 4x and 10x. In this case, what we're going to do is acquire ZStack. So that's acquiring an image at a varying height level, so because we may not be able to get the whole structure in focus with a single image. So just showing on the top, and this is a wide field platform, so we're capturing all of that light, including the out of focus light. Typically, this is a confocal application. And just showing as you're stepping through the sample, different parts of it get in focus while at a different Z-plane or a different height. And then at the bottom, what we've done is we've applied deconvolution to these 3D data sets. And what this is doing is just, very simply put, taking away some of that out of focus light using a software algorithm. So we can approach confocal results with just a wide field platform. Right? And then within the software as well too, so for 3D analysis, this is something that these researchers are also interested in. And I'm going to show a simple example of what you'd be able to do. Using the 3D data set, we're able to reconstruct this data into a 3D viewable, into a 3D render, able to do some visualizations such as rotating the object to get a better idea of how the different structures are within this 3D space. And then we'll be able to do some simple quantification. All of this is within the Mehta Express software that also runs the instrument. Okay. In this case, what we're doing is we're identifying pixels of interest. And then in this case, we're not interested in the central glob of dead cells. We more so wanted to count these cells along the periphery, so we'd be able to perform a filtering step to exclude large structures or very small structures that could be debris. Okay? And then in this case, these are the objects that I'm measuring. And there's multiple outputs that we'd be able to get, not just counts, also such as volume, intensity information. If there are multiple colors in this experiment, we'd be able to calculate colocalization as well, which is something that's important to researchers doing this type of work. And then just showing an extension of this type of approach, which would be monitoring these changes over time. So this would be imaging the spheroid on day 1, and then just showing the comparison 2 days later. So just showing that difference, and then just showing another example. So you can see that we are able to stick with the same sample and then image at the same multiple time points later. Okay. A little bit about the hardware. So I've been talking about the ImageXpress Micro XLS platform. So it's about the size of a benchtop instrument. We have an example over at our booth. You can stop by. It was custom engineered for speed and for image quality. So you'd be able to get about 50,000 wells per day depending on your application. It uses a combination of laser autofocus for plates as well as an image-based autofocus for speed and flexibility. And within the system itself, you could have 4 objectives that you could configure yourself, as well as 5 filter cubes. And as you can see, sample flexibility, we're able to do anything that's pretty much in the footprint of a microplate, so 96, 24 6-well plates up to 1536, and then we can also accommodate slides as well. I mentioned being able to approach confocal, being able to take some of that out of focus light for 3D applications in the software. This is after we've acquired the images. There's also what's called a digital confocal option for during acquisition. We'd be able to increase your contrast while we're acquiring those images, while we're setting up our experiments. So a couple of features. One is the 16-bit scientific CMOS camera. Some of the benefits to this over typical CCD detectors is going to be you have an increased dynamic range going to 16-bit, as well as an increased field of view. So these typically have about a 3.5 times larger field of view than a CCD camera. So that means you're going to be able to capture more objects in a single image, reducing the need for stitching and tiling. And then the 16-bit, the increased dynamic range means that you'd be able to, in the same image, detect low and high expressors. And then a solid-state light source. So this is gaining popularity in the field these days, and so this is over bulb-based systems. It just means a reduced maintenance needs, more uptime imaging, a longer lifespan. Okay. A couple of options for the ImageXpress Micro. So one is environmental control. So this is going to enable these long-term live cell assays by controlling for carbon dioxide, humidity, and CO2, or in temperature. You're going to be mimicking an incubator environment. So we've had some customers that are maintaining cells inside the instrument for up to 7 days. Transmitted light, so you'll see a transmitted light tower here. So this will enable you to perform label-free imaging, or you just want to pop your plate in and observe and monitor your cells. And then fluidics. So this could be used for fast kinetic reactions where you wanted to add a compound and immediately view the effects on your cells, or you could use this for any type of media change during a long-term experiment, for example. Okay. So I talked about the scientific CMOS camera and the solid-state light source. Just a couple of pointers, bullet points about that to reiterate what I said before, which is about 3 times the number of objects in a single field of view. And you can enable whole well imaging. I've got a couple of examples of that in a later slide. And then solid-state light source. So this means increased uptime because of an increased lifetime. So typically about 10 to 15,000 hours versus about 1,000 for a metal halide bulb. And then an electronic shutter versus a mechanical shutter. So all of that leads to decreased maintenance needs. Okay. Oops. Sorry. Oh. And sorry, one more thing to mention would be just flexibility around the light sources as well too. So we have flexibility outside of the standard white light, solid-state light source to include if you wanted to do UV imaging or near-infrared. So that would include the size 7 wavelengths. Okay. So I mentioned capturing whole wells. So this is something that our customers have been very excited about the new platform. Being able to use this for. So at 96 wells, you'll see that you can get a whole well at a 2x objective. So typically, you would need maybe 4 images at once to capture the whole well in 96 well plate. At 4x, you're able to capture the whole well of a 384 well plate. And then at 1536, so this may not be applicable to everyone, but for our high throughput customers, this has been very helpful, being able to acquire the plate as a 384 well plate. You can extrapolate this to say, with the 384 well plate at a 2x objective, you'd be able to do the same thing, which is capture 4 wells at once. All right. Also, this stage precision is very helpful here for objects and structures that extend beyond a single field of view. So I'm going to focus in on this cell in the top left. Say you had processes or tissue slices. The stage precision allows us to be able to tile these sites together without needing to acquire with an overlap, which is typically how larger structures are captured, so that they can be stitched together. And this increases your software processing time. So just showing these And with a zero spacing or zero overlap, we're able to still connect these processes together. And then I mentioned previously digital confocal. So I mentioned a couple of options that we could, with 3D data sets or data sets with a lot of background or out of focus light in them, we would be able to remove that in post-processing with 3D deconvolution. We're also able to remove that while we're acquiring. On the left is an example of an image taken normally without the deconvolution turned on, and then on the right, you can see that these structures now are much more in focus. There's a higher contrast. So an added benefit of this is being able to use a shorter exposure time as well. So for those that are interested in throughput as a benefit, it's an added side benefit. Okay. Let's talk a little bit about the software. So Mehta Express is based on Metamorph. If any of you are familiar with that, Metamorph is the backbone, and what Mehta Express adds on top are plate utilities as well as integration with a database for larger data sets. Setting up an experiment just takes a few clicks, and you're able to save these for use as a starting point later on if you had a predefined acquisition protocol. And then once you've acquired your images, there are several analysis tools available to you depending on your needs. So what this is doing is just showing you an example of acquiring a plate. So it's a 96 well plate with zebrafish actually inside them, just showing how the system's going to look while on the software while it's acquiring the images. So it's building essentially a virtual plate here, and then once it's finished acquiring, we'll be able to review our images. And seeing the plate at this high level allows us to really be able to pick out any types of discrepancies. And so, for example, if you had cells, you'd be able to tell if there's some type of cell distribution issue or whatnot. And these are low resolution thumbnails, so clicking on any of these will pull up a high resolution image to review and take on to your analysis. And then just talking about your analysis options. So in order of ease of use to power and complexity, we have application modules. So these are pre-configured analysis routines for common biological applications. And then on the right, we have what's called journals. So this is similar to a macro in Excel, stringing together different commands and then automating that process. In the middle is what we have a recent release called Custom Module Editor, and I'll talk about that in a couple of slides. But just showing you the range of application modules that are available. So on the bottom is the name of the application module, and on the top is the typical biological application. So it's not set in stone which one you would need to use, but just to show you what our customers have previously used these application modules for. And then just while you're setting up an analysis, just showing you that you get real-time interactive results. So on the top, you get a graphical result, and on the bottom, you also get a numerical table. And those are linked. So we have these yellow cells are highlighted, and that's reflected in the numerical table, and that happens vice versa. So this allows you to get individual object results immediately. Custom Module Editor adds on to that by being able to add either pre-processing or filtering steps. If you wanted to use an application module as a step, but for example, I wanted to remove debris, or I only wanted to analyze a certain population of these cells, I'd be able to do that within the Custom Module Editor. And the workflow is very similar. We'll be, in this case, we'll be teaching the software what we're defining as objects. We're using a click-to-find tool, and it gets selected. And also the previewing our analyses are also interactive here as well. We'll be generating a graphical mask and then also a numerical table that we'd be able to refer to. Once we've run the analysis over the whole plate, now we can get also a visual QC of how the results are by configurable heat maps. So a lot of the workflow is we have an idea of how our cells or how our plate is going to react, or how our samples are going to react to our treatments, and we want our images to confirm that. When we're setting up our analysis, we want the mask, or what the software finds, to reflect what we saw in our images, and then on a larger scale, always confirming back what we saw once we run it over the whole plate. We want to make sure that we're able to identify our controls very easily. That means the software was able to, and if there's any type of trends, we'd be able to see that as well in the numerical results. All right. And then just in summary, so I talked about the cancer cells and spheroids, and using the combination of labware and instrumentation to enable high throughput screening, or getting higher throughput imaging, or getting more information out of it, or ease of use. And just some experimental results, such as uniform size could help with the consistency of your work. And then also that's enabled by the labware and the instrumentation. And then also, about the platform, you'd be able to image, multiple sample types, from beads, cells, tissues, whole organisms that I showed, as well as complex cell culture situations. And then, the software and the hardware, it's flexible to be able to acquire all of that data and analyze it also on the same platform.